Okay, so I hope then Zoom is smart enough to scale everything so that it fits everybody's screen. Okay, so
Right.
I can stop
Yeah,
sharing.
thank you. You tell me when, Ken.
I think we're set. We've got a good group. So join when we begin whenever you'd like.
All right. All right, good afternoon, everyone. On behalf of the Association of Biomolecular Resource Facilities and the Light Microscopy Research Group, I want to welcome everyone to this workshop entitled Fluorescent Imaging Agents in Biomedical Research. The co-organizer of this workshop is Armandi Amer, Arvidas Matiukas, and myself. As you are aware, fluorescence is at the base of a lot of different biomedical research, and this covers the gamut of some of the most abundant or most populated areas of the ABRF, including imaging, flow cytometry, and sequencing. So it is very important, we think, that people who work in shared resource facilities are fully aware of what that fluorescent means, what it is, where it's coming from, what are the characteristics of fluorescent molecules that are going to impact their usefulness or not for the different kind of fluorescent-based assays that we have to perform on a daily basis in our research laboratories. So one part of the workshop is going to be dealing with those basic aspects. The other part of the workshop is going to go a bit on the other end and getting out of the basics and go back to something more specialized. And part of that is that fluorescence has been with us, well, fluorescence has been with us forever, right? But we've known about fluorescence for about 250 years or so. And there are very good fluorescent molecules out there that have been known for a long time, including fluorescent, shown in here, dyeing the Chicago River in Chicago for St. Patrick's Day, or quinine, one of my favorite fluorescent molecules. However, that doesn't mean that you should always stick to fluorescent in the form of typically referred as FITSI for your imaging needs. I will argue that the recent developments in the generation characterization of fluorescent molecules make it imperative that you do your homework, and instead of getting a secondary that's been sitting 20 years in the laboratory freezer and use it just because it's there, you help yourself but by going in something that is better. Dyes are being produced on a continuous basis, that are brighter, more stable. They have specific properties that make them serve as not only beacons, but also sensors of the physiological status of the cells. And those will be dealt with during this workshop. So in this first day today, we're going to be talking about an overview of fluorescent probes by Arvidas Matiukas, and Jason Kilgore will present ways in which those precious 3 photons that you're able to get out of each molecule are really preserved so you can get your signal to noise ratio that you need. And Kelly Lunston will talk about the application and limitation of long stop shift dyes. That you will see they have some very specific characteristics that make them very desirable. On Thursday, Leslie Lowe will talk about sensor probes that can sense the environment of the cells and not just act as beacons. And Luke Levis will talk about new fluorescent probes, and that goes towards the development of new dyes and new resources for, in his case, imaging, but can be applied to any kind of fluorescence assay. And we'll close the workshop with a general discussion that will be led by Mandy Amar. In terms of housekeeping, the presentations are going to be available as PDFs. We meant to have them ready for you today during the talks, but we blew that up. So they will be ready by the talks on Thursday, and certainly they will be available to all of you soon, probably right after this, the workshop session today. Silence your phone, your mics during the presentations. If you have any questions or comments, post them to the chat channel, and the moderators will keep track of them and push them to the speakers at the end of the talk. And remember that after the second talk on Thursday, there will be a general discussions in which you can then save your more general questions or questions that have not been dealt with during the workshop and your interest. Without further ado, I'm going to introduce and see the mic or the Zoom to Arvidas Matiukas, who is our first speaker and the director of the Neuroscience Microscopy Core at SUNY Upstate Medical University. Arvidas has a long experience in both the microscope side of the imaging and the molecule development side of the imaging, and he's an active member of the ABRF and the Light Microscopy Research Group. Without further ado, Arvidas, the floor is yours.
Thank you for the introduction. Okay, so let me start my PowerPoint. Okay, and now I need to start sharing it.
While Arvidas gets his presentation going, Mandy posted the talks in the chat channel so you can download them right away.
I hope the screen sharing is working by now. Yeah. Okay, so let's start. So my talk is overview of fluorescent probes, and there are many aspects, but I mostly concentrate on the ones that are within my main field of expertise. And I would tell this as biophotonics, which is the most well understood and well quantified. And here the aim is to provide fast and efficient diagnostics that is compatible with live organisms, that covers wide variety of sample types and sizes, ranging from subcellular to whole organism, and a scale size on the order from micron to tens of millimeters. Also is related to different imaging techniques and devices as microscopy, spectroscopy, flow cytometry, and even sequencing, as Guillermo mentioned earlier. Why we are using the fluorescence? Though it is relatively old, was first observed about 150 years ago, but by now it is preferred method for labeling and providing high contrast for biomedical images. Also, it provides good resolution, provides quantitative measurements, allows not perturbing imaging with some limitations, also allows different type of either fast or long-term and even high-volume experiments. I briefly mention that there is alternative to fluorescence. There are different modes like polarization, second harmonic generation, and Raman scattering modes that do not require any dyes on the sample. Now let's look at some basic properties of fluorescence. So on microscopic scale, any atom or molecule follows quantum laws and has these discrete energy levels, or if there are many of them, these become energy bands. So as you see in this Jablonski energy diagram, a lot of processes are going on, and they are very fast and very short. But let's concentrate on the most important, which is within this oval shape. So under excitation, photons, electrons jump from ground state into the excited state. Then there spontaneously may happen some internal relaxation, internal loss of energy, and finally, spontaneously electrons fall back into ground state, emitting longer wavelength photons. So as we see, the processes are very fast. There is energy loss between excitation and emission photons, resulting in Stokes shift up to 100 nanometers, and the photons may be emitted at different direction and polarization. Now, because practically we always deal not a single molecule, but with many of them, so on macroscopic scale, we need to pay attention to following properties. That fluorescence is a cyclic process, so it is like a continuous fountain. And typically, until the fluorophore fluorescent process is killed, we will talk about this later, for example, by photobleaching, it repeats on the order of 10,000 times. So as we see, on macro scale, it produces absorption and emission spectrum. They may have a single peak and may have several peaks corresponding to the transition between different states. Also, fluorescent produces time spectra. Not all photons, they are emitted at the same time, but there is a delay, and fast electronics these days allows to measure and plot these time spectra that can also be used for different applications. Then the property is that emission can be saturated because these energy bands are like shelves with limited capacity, and they may become full, and then further increasing excitation does not increase the emission. And finally, fluorescent is environment dependent. It depends on the solvent polarity, pH of the medium, and nearby molecules. Specifically, when looking at the fluorescent probe, we should pay attention to the following properties. Excitation spectrum, which can be quantified by peak wavelengths and width. Extinction coefficient, which shows absorption efficiency. Emission spectrum, which again we can quantify with peak wavelength and width. Then quantum yield, which shows the efficiency between excitation and emission photon conversion. Relative brightness, which theoretically is product of the previous two mentioned parameters. And there are, I forgot to put a reference here, but if somebody interested, I can tell later. There are tables that quantify relative brightness in the scale of 10. So, and roughly the relative brightness can differ about 10 times. And finally, photobleaching, which is a distraction of this fluorescence cycle. Here, I added interesting picture, which shows what happens if we excite not at the peak of the excitation spectrum. At other wavelengths, the fluorophore still is excited, but less efficiently, which means that emission spectrum is has lower intensity. So as we see, excitation at peak produces highest intensity, off peak, lower intensity, and at secondary peak, so low position of excitation curve also produces low emission. And again, as I mentioned, these properties generally are environment dependent. And for example, a warning when looking at spectra and excitation and emission peaks, make sure that they were measured under physiological conditions, because quite often the spectra are measured in spectrometers, and various non-physiological solvents can be used. Of course, there to be used to their best, these properties must be compatible with the imaging system you will be using. Next, in a practical application of the fluorescent probe, we need to pay attention to experiment design, probe selection, probe acquisition and storage, specimen labeling, and imaging. And ideally, these steps need to be reiterated, because as I mentioned, except the photophysics, the list of chemistry and biology of fluorescent probe is not that exactly understood. So several approximations or iterations typically are required by the trial and error to get the best results. And of course, always it is advised to start with tested design or protocol. And regarding the protocols, also be careful about protocol reliability and reproducibility. There are, I quote here, 2 papers. By the way, author of the first one is the co-organizer, Guillermo Marquez. You can ask him additional questions about that. So in experiment design, we should look in specimen type, then what we want to study and label, structure or process or even function, look at the available imaging systems, and then select the probes and try to identify the optimal probe. Additionally, we may want to look at properties of specimen as well, which can be live or fixed, of different type and size, have different target concentration that we would like to label, have autofluorescence, provide different probe localization, probe retention and metabolism, probe mediated cytotoxicity and phototoxicity, which is important for the live samples, and quenching, which is like media produced killing of the fluorescence. All these properties also affect your final result. There are some procedures that can enhance your specimen, such as enhancing optics by matching mounting medium refractive index, then clearing the sample if you want deep imaging, expand the sample to improve resolution, apply anti-bleach and anti-fade agents. There will be a separate talk about that. So with these enhancements, we can compensate imperfections of the probe and imaging system. Here I briefly list the major types of fluorescent probes. They can be organic dyes that typically are used for structure labeling, can be Q dots that are non-organic. They are very bright and very bleach resistant. There are also special application probes such as multiphoton photoactivation, super resolution imaging that may specifically require these specialized probes. There are environment probes for sensing membrane voltage, pH of the medium, ion concentration, some proteins, and of course, fluorescent proteins that can be genetically engineered, and hybrid probes that can be a combination of protein and organic dye. Now, speaking about final result, we would like to get the strongest fluorescent signal. So as this formula shows, it depends on excitation, probe concentration, probe absorption, probe quantum yield, and detector properties such as emission bandwidth and efficiency of detection. In practice, just high number of or high value of fluorescence signal may be not good enough if it is not over the noise, and ideally it should be much higher than noise. So to optimize these values, we should try to optimize each step. So for example, while labeling with organic dyes, we should take care of the storage conditions, as Guillermo already mentioned. So 20-year-old probe, I mean, not designed 20 years ago, but sitting on the refrigerator shelf for 20 years is definitely not good. And what other labs may lend you also may be not good enough so that they gave you. We need to pay attention to solubility, loading concentration, permeability, loading conditions, washing the dye, its acute and phototoxicity. Also, we may take advantage of antibody-mediated protein labeling. There are several protocols. These, of course, properties of the probes need to be compatible with the specimen, so please follow and modify as needed existing protocols. And here, our goal is to achieve maximum label density without significant perturbation of the specimen. So briefly, types of labeling can be either chemical bound or other as antibody-mediated, genetically engineered fluorescent proteins, or hybrid type. Now, briefly, if we look at the optics of the probe application, so when optimizing excitation, we should look into not saturating excitation states, matching emission excitation spectrum with source wavelength. Preferentially use white light laser, where we can continuously adjust excitation wavelengths and have exact match. Or as alternative, use broad spectrum source with a matched filter. Blank the excitation when not imaging or observing. Minimize excitation duration. There are some studies that show that the damage of excitation is nonlinear, and short pulses are preferred because between these pulses some repair mechanisms may be employed. And also use anti-fading, anti-bleaching agents. So available options among listed may depend on your imaging system. On the detection stage, we can optimize by matching emission filter, matching dichroic filter, which is essential to separate excitation and emission, because emission is much weaker than excitation. We should look into optimal exposure time to optimize so that the signal to noise ratio is at least above value of 3. We can use hardware and software averaging to decrease noise. We can use pixel binning, and we can use internal detector amplification, which also allows further optimizing signal to noise ratio. The listed options typically are instrument dependent. So the role of imaging system is to provide efficient excitation, emission collection, and emission detection. Here I listed several most popular system types, such as wide field, confocal, multiphoton, superior resolution, light sheet, microscopes, flow cytometer, and also fluorescent probes are used in some sequencing systems as well. Now, in practice, we use several probes or several labels simultaneously because we want to address our specific research questions. So in that case, we also need to take into account the distance between excitation emission peaks, which for the best results should be about at least 100 nanometers. The spectra should have minimal spectral overlap. I will show in the next slide special software, the spectral viewers and databases are available for that. Then we need matching probes to the available excitation sources and filters, and the availability of sources can be laser, LED, or traditional mercury light source. It is advised to equalize as much as possible probe brightness so that I'm sorry, I mean not exactly brightness, but fluorescent signals of different probes, so that it is recommended to use brightest probe for the least abundant target. So we also should consider mixing different types of probes, and regarding the maximum number of probes with the current imaging system, 3-4 probes are okay. More than that, 5 or more can run you into problems, mostly with the crosstalk or the bleed through, and additional measures need to be taken. Now, regarding multicolor imaging, we should care about matching emission spectra to existing emission filters or using spectral detector. Choose between sequential or simultaneous imaging of the fluorophores. Use software and mixing for crosstalk compensation, and optimizing components of excitation, emission, and detection so that the product is gives you maximum value of the signal. So here is an example of the spectral viewer, which allows you to visualize, for example, what you see here. The curves are listed here. So we have excitation and emission spectrum. So in as these reddish colors for the probe site 2, and the green curves are for site 3. We see some partial overlap, and the bandwidths of the excitation spectra, for example, here for site 2 and site 3, are selected so that only that there is no cross excitation on a single probe is excited at a time, and the emission bandwidths are also this curve and that curve are selected with no overlap. Additionally, for the quantification, you can add the spectral characteristic of your light source. Here, this curve is for the LED light source, and this curve is spectral sensitivity of your CCD camera. And also, quantitative calculators are available where all these spectral and optical properties can be quantified. You enter them, and the system calculates your fluorescent signal and signal to noise ratio. I would like also to mention some advanced methods, such as signal separation by emission timing. As I mentioned at the very beginning, that the fluorescent photons, they have different timing, and different probes can have very different spectra, and this is realized in the method called FLIM. Some signal separation as well can be done by polarization of the emission photons. There are methods and protocols that allow sequential probe loading and removal up to several tenths of probes. Then also I would like to mention ratiometry methods based on spectral shift. Specifically, here is a picture that shows for the Fura2 calcium sensor. As you see, the spectral curves, they shift in opposite direction around so-called isosbectic points, where the spectral, concentration sensitivity is 0, and taking ratio between this wavelength and that wavelength gives us a value of the ion concentration, which is self-calibrated.
Aaron,
and
you're down to 2 minutes, okay?
Yeah, thank you. And similar principle can be done as a ratio to emission wavelengths. Why this self-calibration is important? So if you recall, the final signal is product of different factors. So besides the probe properties, environment factors, and excitation emission values may have variation in time or in space. And self-calibration methods allow to overcome this and get exact results. Okay, now briefly about some limitation and problems that we may run into. So photobleaching and phototoxicity limits fluorescent signal. So we can mitigate that by selecting fade-resistant probes, labeling densely, using anti-fade, anti-bleach agents, and minimizing exposure. Also important is minimizing noise by reducing autofluorescence. For example, by bleaching, this won't work on the live samples, by reducing background and reducing detector internal noise, for example, by cooling it. So here briefly I mentioned, listed some obvious probe sources and as well application protocols sources, and want to point out that achieving best results requires synergetic knowledge and experience in diverse fields of physics, optics, chemistry, biology, medicine, and engineering. So here is some literature of interest that you can take a look about details, because these handbooks are like 1,000 pages, and I tried to pick up most common and most important moments. So in summary, the take-home message is, what is good probe? Good probe is the one that matches your labeling and imaging needs, and which is applied and imaged under optimized protocol. So thank you, and I am looking forward to questions in the chat, or here is my email. You can contact me later and privately.
Thank you very much, Arvidas. We are running a bit tight on time, but I have a quick question for you. Is there any relation between a Stokes shift and the lifetime in fluorescent molecules, or are they entirely separate characteristics?
Well, okay, let's. So Stokes shift depends on the internal energy loss, which is like the distance between different excited energy bands. While the transition times, they are more complicated. They are governed by quantum laws and by different quantum characteristics of the electrons on these energy levels. So I would say that there is no direct relation. And why this is, by the way, the basis of these FLIM methods, that probes can be fluorescent signals from different probes can be filtered not only by their fluorescent spectra, but also by their timing spectra.
All right, thank you. I think we're going to be moving on because we started a bit late, but remember that on the second day, there will be at least 15 minutes, maybe more, for Q&A, and we hope all of the speakers will be available. Also, you will have an opportunity to check Arvida's presentation, the PDF, that so you can also be look at the slides and maybe come up with some questions for Arvidas. All right, thank you very much, Arvidas. Do you want to go ahead and introduce our next speaker?
Yes, just a moment. Let me shift. So our next speaker will be Jason Kilgore, who has worked at Thermo Fisher Scientific and former Molecular Probes for 22 years as a specialist in fluorescent dyes and conjugates for use with microscopy, including different antibody reagents, organelle stainings, and anti-fade mounting media. He started in research and development, developing these products, including leading a research group for many years, then moved to a role as a technical support scientist. He attended Hendrick College at Arkansas, Central Michigan University, and Oregon State University. So he currently resides in Eugene, Oregon, and enjoys spending time in mountains and coastlines of the Great Northwest.
Thank you, Arvidas. I guess I can share my screen here. So,
yes, I'll stop.
Yeah, so, very good. There we go. Sorry, I'm trying to figure out how to get to it on the system here. One moment. Ah, there we go. Okay, so you should be seeing my screen here. So thank you to the organizers, all of you, for organizing this, and thank you for the introduction. So, okay, so what's the point of a fluorescent dye? It's all about, as Arvidas pointed out, signal to background or signal to noise, right? Getting the strongest signal above background. You can think of your dye as a beacon in the night or a spotlight against that background. And so you, as Arvidas pointed out, you choose a dye with the greatest quantum yield, strongest excitation coefficient. But what's the point in getting such a great and stable dye if it's just going to photo bleach away, right? So in this presentation, I'm going to talk about some practical things that you can do to reduce photo bleaching, as well as anti-fade solutions. And some of you who have a lot of experience with microscopy, some of this will be basic, but stick around to the end because there will be some information on a newer product that you may be greatly interested in. Okay, so what is photo bleaching to begin with? Basically, anytime you shine a very strong light, whether it's the light source on your microscope or sunlight onto a molecule of a dye, it causes loss in molecular structure, an actual breaking of the covalent bonds, but also an increase in the movement of the molecule. Fluorescence for many dye molecules requires a rigid structure, and if you get a lot of movement in the bonds there, it causes a reduction in the fluorescence intensity. And in this presentation, I'm not going to go into the theoretical aspects, first order, second order photo bleaching or the mathematics. It's all going to be empirical here. And then in addition to the actual molecular structural loss of the dye, there's also light exposure can create free radicals and singlet oxygen, which can further degrade dyes by moving through the media that your sample is in. So in order to reduce the photobleaching, you therefore have to reduce the light input into the sample that can cause the molecular structural loss, and put in agents that can sop up the free radicals and singlet oxygen. So what does photobleaching look like? Well, here on the right, you can see an example image of a cell labeled for tubulin in green and actin in red. And with continued exposure of light, you actually have a dimming effect of the dye. This is actually quite long exposures, 5 seconds and 15 seconds on some Alexa Fluor dyes. And on the left, you can see an example plot of some of these dyes. Usually the x-axis is in time in terms of milliseconds or seconds, but you can also plot it based upon number of scans, as this one is. But you can see that there's it's normalized to 100% on the y-axis, but there's an initial drop for many dyes, which is sort of that first order kinetics, and then a slower reduction of intensity over time. Again, here's sort of what it looks like in terms of intensity loss over time. In the bottom row there, you can see with no anti-fade components, these cells at fairly low mag, 20x objective was used. With no anti-fades, just a PBS and glycerol, you get an initial intensity, but it drops to almost nothing within just a few seconds. Whereas if you add an anti-fade component, like at the top there, you have prolonged diamond, you can maintain the intensity over a much longer period. There'll still be some dropping of intensity, but here by eye, at least, you don't see much. Of course, the higher the magnification, the worse it will be. So causes of photobleaching and things that you can do about it. First off, you want to choose a more stable dye. Many of the traditional dyes dating back decades, such as fluorescent, rhodamine, coumarins, and pyrenes in the blue, those tend to be much more photolabile or prone to photobleaching. So over the years, companies like Molecular Probes have developed better dye options with some of those factors that Arividas was talking about, right? With better quantum yield, better extinction coefficients to give you a brighter emission and a more efficient absorbance. But also changes have been made in the structures of these dyes to allow them to be more photostable. So for instance, fluorescent was derived to add some fluorines to it. And this fluorination became Oregon Green 488. And as you can see on the graph on the right, it had a little bit more stable photobleach curve. And then I think it was in the mid-90s, we released Alexa Fluor 488, which was a sulfonated form of fluorescent. And by adding those sulfate groups, we were able to get a much more stable dye, as you can see in the graph at the top. Of course, this sulfonation also added other properties. It was a bit brighter and less pH dependent too in terms of the intensity, whereas fluorescent, for instance, you get a reduction of intensity at lower pHs. Matter of fact, it can be used as a pH indicator for that reason, whereas Alexa Fluor 488 is still stable in intensity down at lower pHs like pH 4. So these derivations can be important. And over the years, there have been dyes introduced by other companies too. Of course, I'm biased being from Molecular Probes, and we have those Alexa Fluors going from blue all the way up to near infrared. You can see some curves up there, but there are many good dyes out there, such as the Dye-Lite dyes from Pierce, which is now part of Thermo Fisher. The Psi dyes that you're probably familiar with, Psi 5, Psi 7, E-Fluor dyes. There's a lot of different options out there, and almost all of them are very stable compared to those traditional dyes. So choosing a more stable dye is probably the best thing that you can do. But it's not always a possibility. You know, sometimes there are certain derivatives of fluorescein that you have to use for whatever reason, or maybe the only options available out there for a conjugate might be fluorescein. So another thing that you can do is have a counterstain that's more stable. DAPI is a very obvious one, or Herx 33342 for live cells. You can label your nuclei and then use that wavelength to find the field that you want and get into relative focus, and then quickly switch over to the photolabile dye, like in green, and then snap your picture. So for instance, I have an example there with some fluorescein labeled tubulin and DAPI. So you image in DAPI and then switch over to tubulin and take that picture. Another cause of photobleaching has to do with the settings that you're using and the environment that you're working in. Ariv just mentioned how you want to optimize your settings on your imaging system, but this is also true not just for getting the best signal to background, but also for reducing photobleaching. For instance, the best thing you can do is limit the light exposure. So the intensity of light that's hitting your sample. In our EVOS microscopes, for instance, we have light cubes with LEDs as the light source, and you can change the voltage or intensity of those LEDs. So if your dye is photobleaching, you want to reduce that intensity and maybe increase the gain setting to get a more efficient capture from the detector. For confocal systems, you can lower the laser power and voltage. The trade-off, of course, is by limiting your exposure, you're also limiting the signal to background, right? So that's always a trade-off there. For standard wide field systems, traditional ones often have a mercury bulb, so you can't really adjust the mercury bulb intensity. So instead, you'll use neutral density filters. These are smoked glass filters that you can slide into the light path of different percentages. So when you're setting up your sample, trying to find the right focal area, or focal depth, you'll slide those in, and then when you're ready to take your image, you slide them out and snap your picture at the right exposure time. Exposure time also is an issue. As the name implies, the amount of time that you're exposing your sample to snap your picture. Reducing the exposure time will reduce the amount of time your cells are being photobleached. Also reducing your signal to background, so it's a trade-off there too. And of course, reducing the time you're just visualizing by eye. The more you're scanning around your sample, looking for the area of interest, the more you're photobleaching those areas that you're looking at. So sometimes it's important to be fast about it. The magnification that you use matters. So the higher the objective, the higher the numerical aperture you're using, the more efficient light exposure you're getting on your sample, and therefore the more photobleaching that you're getting of your dye. So if your assay allows for it, sometimes you can go to a lower magnification to capture your image. Or if you happen to have multiple objectives around of that magnification, choose one with a lower numerical aperture to lower the light. Of course, the lower numerical aperture also means that you're collecting less light on your sample and therefore maybe less resolution of your specimen. So that's a trade-off as well. For confocal systems, your laser rasters across your sample like a line, and that rastering can be controlled. So it has a certain speed of raster. The faster the speed, the less photobleaching, or the lower the speed, the more photobleaching, so you can change that speed. Also, the raster goes over more than once. You can change the average number that it rasters over a line. The higher the average, the more times it passes over those molecules and the more photobleaching you'll get. So to reduce the photobleaching, increase the speed and lower the average rastering. But the trade-off, of course, is intensity of your sample and the resolution of your sample. Also, it's sort of basic, but keep your dyes out of sunlight. So don't work right next to a bright window, and keep your samples covered loosely with aluminum foil if they're sitting on the lab bench. Lab lighting isn't really much of a factor for most dyes in terms of photo bleaching, so you don't need to turn the lights off or anything. Sometimes we get that question in tech support, and we're like, no, 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 you don't need to turn off the light. And if you're storing your samples in a refrigerator, make sure it's not glass fronted unless the dye is in a box or covered with aluminum foil. Also, free radicals or reactive oxygen. When you expose your dyes to light, you're generating these species, especially singlet oxygen, and this will interact with your dye molecules to speed the degradation. The best thing you can do to reduce this, at least in fixed cell samples, is the use of an anti-fade solution. And now there is a live cell or a couple live cell options as well. Anti-fade solutions will have antioxidants and free radical scavengers that will remove those free radicals. Also, by using a curing or hardening mounting medium, that hardening effect slows the kinetics of the movement of those free radicals. And then lastly, keeping your sample cold, especially...
Eastern.
We have some background noise, thank you. Keeping your samples cold can reduce the kinetics as well. So if you're using a hardening mounting medium, once it has cured at room temperature, after a day or so, then you can store your slides at 4 degrees or even -20 to slow the off rate of your sample, but also to reduce the kinetics of the movement of that ROS species. So let's turn now to a quick discussion of anti-fade mounting media. I had the honor of developing some of those and still advise our R&D groups on that. What is a mounting medium? That's just the solution that goes between the slide and the cover slip to bathe your sample in. It's not always an anti-fade. An anti-fade is one that has anti-fade components, free radical scavengers added into it. But sometimes we can say mounting media to mean something that's not necessarily under a cover slip, but also what you would put in a 96 well plate for imaging on an inverted microscope, or that you would bathe a whole mount or spheroid in. Traditionally, the mounting media were solvent-based. If you go back 50 years or even 100 years, I think some of the original mountants were resins, sometimes even derived from tree resins originally, that were dissolved in toluene or xylene or other organic solvents, and the really old ones would yellow or crack. Modern versions are clear. And include Permount, for instance, or DPX, which stands for distyrene with a plasticizer and xylene. Cytoseal 60 is another common one from Richard Allen Scientific. Here at Thermo Fisher Scientific, we sell Histomount. All of these are great for traditional chromogenic dyes like H&E, hematoxin and eosin, DAB, and others, mainly on paraffin sections. These solvent-based mountants, you have to run your sample through a graded ethanol series to dehydrate it and then into a solvent before mounting. And this is potentially damaging to some samples like cells or cryosections. It can cause changes in morphology. And if you use fluorescent dyes with them, they can even extract fluorescent dyes if they're already on the sample or degrade fluorescent proteins. So a lot of these solvent-based mountants aren't really ideal for fluorescence. Some of them have autofluorescence like Permount, which will drastically lower your signal to background ratio, and you don't want that. And some of these, like Cytoseal or DPX, will quench fluorescent dyes to some extent or even completely in a few cases. So instead, you'd want to use an aqueous-based mountant for fluorescence whenever possible. An aqueous-based mountant is one that's, instead of being in an organic solvent, it's in an aqueous buffer, usually with glycerol as a plasticizer. And because it's aqueous, you don't need to move your samples through a dehydration series. Instead, you can just tap off any excess buffer, put on a drop or two of your mountant and lower your cover slip at an angle onto it and image, or in the case of curing mountains, let them cure or harden before imaging. Many of the aqueous-based mountains now have anti-fade components in it. Examples of anti-fade mountains include our Prolong and Slow Fade anti-fade media. Others might be Fluorescent G. Vecta Shield is a very popular one out there from Vector Labs. These are formulated in a way that won't quench or minimally quench most fluorescent dyes and will have an anti-fade component in them. Some of them do have some drawbacks. The traditional Vecta Shield, for instance, has some blue autofluorescence, which goes away quickly and can quench far red dyes. So I'm going to discuss some of our fluorescent anti-fade mounting media, not as an ad for our company, but really to discuss some of the aspects of anti-fades and what we've done to develop them over the years to make them better. Really quick, curing mountains are ones that polymerize or form a matrix of polymer or plasticizer, usually through a dehydration. So when you put the sample under the cover slip, you let it sit, and the water in the buffer will evaporate off and leave behind a matrix. Now, the solvent-based mountains often harden to the point where you'd have to chip them off. But with aqueous mountains, it's more of a gel, a hard gel like a gummy bear consistency. Once they reach a fully cured state, you get the best refractive index for the best resolution. And also when it cures, it slows the off rate of conjugates and dyes. So that's another reason to go with curing. Non-curing mountains stay liquidy. They don't harden. And this is good if you're going to image right away. So like our slow fade products, if you're going to image your sample immediately after mounting and then discard your sample after just a few hours or a day or two, then you can go with a non-curing mountant. But if you're going to archive your samples for any length of time, I'd go with a curing one. So historically, back in the 80s and 90s, people were making their own anti-fade media and publishing on it. Some of those components that they would add into a glycerol solution were NPG, PPD, Dabco, hydroxyquinone, Trolox, which is a water-soluble vitamin E. All of these had in common that they were antioxidants and free radical scavengers, but they weren't very good. I've tested them. They're pretty poor for most dyes. Some of them are okay for fluorescein. NPG and hydroxyquinone and PPD will actually oxidize and turn brown over time. You don't want that. And then Dabco, for instance, is this noxious component. We used it in our original slow fade. It smells like rotten peanuts. It's really awful. So back in 1990, 1991, we introduced the first commercial anti-fades with slow fade and later slow fade light, which had Dabco in it. And they were okay. They improved the stability of dyes. Vecta Shield from Vector Labs was also introduced around that time, and it was pretty good and still remains quite good. And then a few years later, we released Prolong anti-fade mountant. Prolong was, I think, the first anti-fade mountant that cured or hardened that was commercially available. And it had a proprietary antioxidant that you had to mix with the base, and it was difficult to mix up, and you had to use it right away or it would turn brown as a stock solution. But nonetheless, it was still really good for its time. And we just discontinued it a few years ago. It was still quite popular. But then in 2004, I was part of the group that developed Prolong Gold and Slow Fade Gold. No longer did you have to pre-mix the solution or mix the solutions. Now Prolonged Gold was pre-mixed, and they both slow fade and Prolonged Gold had the same anti-fade components, which were much better for a wide variety of dyes, including our Alexa fluors, but also traditional dyes. The slow fade was much better than, or slow fade gold was much better than the original slow fade. And though these were better options and are still available, they weren't very good for fluorescent proteins. GFP in particular has a problem in that the fluorophore, which is the fluorescent part, is surrounded by this barrel structure of protein. And that barrel structure is thought to prevent many antioxidants from getting to the fluorophore to protect it. VectaShield was okay in terms of GFP photo bleaching, but most everything out there was pretty poor. And so in 2014, we decided to develop our Diamond Prolong and Slow Fade products. We added some additional components to it that now worked well with GFP and its variants and other fluorescent proteins like M-cherry, for instance, and we're able to get better photo bleach curves. For instance, here on the left, you can see Prolonged Gold in the blue was pretty poor, dropped quite precipitously in its intensity, whereas VectaShield Hardset, which is their hardening version at Vector Labs, was okay, and Prolonged Diamond did much, much better. Lower right are the slow fade or non-curing versions, where Diamond and VectaShield were doing very well. And by the way, VectaShields, I mentioned how they had a blue autofluorescence. Their Hardset also had some outgassing, like bubble formation under the cover slip. But they've recently introduced new versions. Vector Labs introduced VectaShield Plus, which is their non-curing, and VectaShield Hardset Vibrance, I think it's called, for the curing. I haven't tested them, but I think they've overcome some of those limitations. I don't know how they compare to our options. Well, then in 2017, we went for the brass ring, if you will, and that is an anti-fade that not only has good anti-fade components for a wide variety of dyes to combat photo bleaching, but also a refractive index like that of glass. Glass has a glass used in microscopy has a refractive index around 1.52, and all the other mounting media out there that we offered, including VectaShield from Vector Labs, is around 1.46, 1.47, and that 0.07 refractive index actually makes a pretty big difference in terms of the resolution that is offered. So that refractive index is important because if you're ever swimming and you stick your finger in the water, you'll see that when you look through the water, it seems displaced from what you see in the air. And that's all about the bending of light as it goes from the air into another media, water, and the refractive index differences and mismatch between the air and the water. Well, this is true in microscopy as well. The light travels through the glass of the objective, but then into air or immersion oil, which has a different refractive index potentially, and then the glass of the microscope slide, which is a different refractive index, and then into the mounting media. And with every change in medium, you now have a different refractive index and a scattering of the light, which reduces your intensity and your resolution. In this image in the upper left there, you can see a bending of the light as the angle of incidence is different from the angle of refraction. Ideally, you want a straight line there where the angles are equal. So in order to get that straight line, you have to keep the refractive index the same for each medium that the light passes through, ideally that of glass. So the glass of the objective immersion oil would be the same as glass. You have the glass slide and then a mounting medium with the same refractive index of 1.52. And we did it. So we developed Prolonged glass and slow fade glass by adding additional components into the base that now gave it a refractive index at 1.52 without reducing the clarity of the solution when it has cured, in the case of prolonged glass, and still has an anti-fade component in it that can reduce the photo bleaching. So on the right here, you can see an example of VectaShield on the left, where you're able to image about 30 microns deep before you start losing the morphology of the nuclei and the intensity. Whereas with slow fade glass at just 0.07 difference, now you can image as deep as 80, 90, even 100 microns with a standard wide field microscope. And that's important. So now you don't have to go to 2-photon imaging or confocal potentially to get that deep, or light sheet microscopy. Now you can use your standard wide field microscope. With that lower refractive index, you also have a stretching of the light. Some of you might know the term point spread function, right? So when you shine a light on a dye molecule, it emits light in the z-axis as a sort of cone up and below the source of light, and that's a diffraction of the light. Well, you have a stretching of that cone, if you will, with lower refractive index. And so here where I compare our prolonged glass at the top to prolonged diamond at the bottom with a microsphere, I think it was a 1 micron microsphere, and you can see a stretching instead of a round sphere. You see a stretching of the light, and the deeper you go in the tissue, 40 or 100 microns, you see it stretches out. With the older prolonged diamond at a 1.45 refractive index, you have a much bigger stretching of that light than you do with the prolonged glass. And that stretching means you're losing resolution in the z-axis. Here also is another example. This is a 100 micron pig liver sample. Prolonged glass versus VectaShield, and again, you're only getting maybe 30 microns deep with the VectaShield hard set versus, sorry, you can't read the y-axis there, but that's, I think, 80 microns for the prolonged glass with DAPI. So much deeper penetrance. And the anti-fade curves still look great. So for FITSI or Fluorescent, they're on the left. You can see that prolonged glass and diamond and VectaShield are all quite good, and GFP on the right also for those, retaining up to 60 to 70% of their initial intensity, even after 50 scans on a laser confocal system. And here's how it looks just on the slide. You can see a slight clearing effect due to refractive index matching of prolonged glass on the left, and the VectaShield hard set on the right had that outgassing or contraction that led to air pockets, which they may have fixed with the vibrance form. And then we released the slow fade glass version in 2019. An interesting thing with refractive index matching, this was sort of unexpected. This is a 1 millimeter thick rat brain. And when it sat in the slow fade glass for a long period, here we're talking a day, even 2 days, you have this clearing effect. Now, slow fade glass is not a clearing agent. We don't sell it as that. We have other products like CytoVista for that, where you clearing agents actually make samples more transparent for deeper resolution imaging, sometimes by extracting fats, for instance, from the tissue. Here, it's just an intercalation of the slow fade glass into the sample, and by intercalating it, it changes the refractive index of the tissue such that it's now fairly translucent, such that you could even read through it. That was a pretty cool thing. And here you can also see a deeper imaging of slow fade glass versus diamond and gold versions. Lastly, I want to talk quickly about live cell anti-fade. Prior to 2015, the only live cell anti-fade to keep dyes from photobleaching in live samples was a company called Oxirace. They have enzymes derived from E. coli plasma membranes, and these enzymes work as antioxidants of the media. And it's used in a number of industrial purposes, but they also released OxyFluor, which was meant for live cell anti-fade. It wasn't very good. We tested it, it was all right. Well, we licensed it to Thermo Fisher and improved upon it, and now we've released that as Prolong Live, where you add it to your media and let it sit for 15 minutes up to 2 hours. And then you would label beforehand, preferably, but you could label after. And it will protect dyes to a large extent. This was released in 2015. Vector Labs also sells Vecticel Trolox, which is a water-soluble vitamin E that has some antioxidant to it. Trolox isn't very good. I haven't tested their product, but I'm skeptical. Of course, anytime you add something to live cells, you risk affecting live cell function. So for live cells, I recommend doing those earlier things I mentioned, limiting the light exposure, throwing in neutral density filters, and changing settings as much as possible. But if those things are not enough, under normoxic conditions, if you will, then you can add Prolong Live to reduce the reduce the photo bleaching. Here's what it looks like, right? So in the bottom, there's no live cell anti-fade, fairly low magnification of some cells labeled with GFP and Herxt. Herxt is a blue nucleic acid stain. And you can see that by the 60th or 100th image, even at this very low magnification, you've lost most of your signal. Adding the Prolong Live allows a whopping 240 images that you can take before you start losing it to a significant degree. And some example curves, you may not be able to read this, but basically in the green there is Prolong Live, and then the red is without anti-fade. In the upper left is GFP, and you can see a slight improvement in the photo bleaching such that it allows you to have 50% more images before it reaches a 50% intensity. And that's pretty impressive, I think. But we're not yet at the point where we've developed one that's as impressive as our fixed cell anti-fade solutions. Then there's a number of other dyes here, RFP at the top in the middle, and then some mitotracker dyes for mitochondrial labeling, and Herx 33342 in the lower left. And you're improving by as much as double your, or even 190% more images by using this. So it can be impressive in terms of collecting enough of samples before they photo bleach to 50% intensity. And also we were worried when we tested these, you know, when you're adding an antioxidant into the solution, how are you affecting the cell viability and proliferation, right? Because these are live cells, you don't want it to be toxic. So we tested a number of kits. We did a traditional live dead cell imaging kit. We did cell viability and proliferation with a Presto Blue reagent on a plate-based assay. We looked at cell proliferation with EDU, where it takes up EDU on cells that are proliferating into the DNA, or a Cyquant kit. But all of these kits showed that there was no measurable difference in terms of the viability or the cell proliferation, even as much as 2 days worth of exposure to the reagent. Not that you're going to do that, but it was a good test to show that at least with these conditions, you're not going to see any effect on cell viability. Now, in practice, you're not going to use it probably to image cells more than an hour or two, I would think, for live cell assays, because there's other effects that can affect cytotoxicity, like CO2 concentration and temperature. Okay. So I think that's pretty much everything to give a basic overview of photo bleaching and things that you can do to protect against it. Of course, if you have, we have some Q&A here, but if you have questions later, feel free to email me at my email address there. Okay, any questions?
Thanks, Jason. There is, I have a question, and is that you're talking about the prolonged live, right? And the solution is to add. And it's been I won't say commonplace, not as common as it should be, to use a special media for live imaging.
Mm-hmm.
And my understanding there was that the emphasis was in removing.
Mm-hmm. Well, for instance, we sell Fluorobright and also a product called Fluorobright is a DMEM option, and live cell imaging solution, a generic name there for a heaps-based solution. Those have removed phenol red, which can quench dyes, and have removed components like riboflavin that can cause autofluorescence in the background. So not only are you not quenching your dye, but you have better signal to background. There's no anti-fade components in those, but you can certainly improve your initial signal by doing those things. You can also use media without phenol red. Every company that sells media sells options for most media without phenol red. But I would still, if your dye is photo bleaching, I would still add Prolong Live or some other live cell anti-fade like that.
All right.
Questions in the chat. Somebody asked in slide 20, did the graph show PBS glycerol as good at preventing bleaching as the new polymerizing mounting media?
No, so the 50%, so for GFP on the right, 50% glycerol was actually not much worse, frankly, than some of the other options there. Prolonged glass was better initially, but by the time you get to your 50th scan, you're losing a lot of your signal. It's not super impressive there under those conditions, I agree.
Know of an example of a cell dye that can be stained a day before the fixation and still be bright?
A day before fixation in live cells? Yes, but the problem is that with live cells, many of the labels that you do will be pumped out over time, like calcine, for instance, is an example, through active efflux of your dye. Most of the things that will bind covalently to the cells will affect cell function, with some exceptions. Cell tracking dyes like Cell Tracker Green and Cell Tracker Blue, we sell the Cell Tracker. Those diffuse into the cell and bind to proteins covalently and are retained long term. So you can image days later, and we haven't seen any significant toxicity issues. So in those cases, yes, you can label ahead of time and then later image with the Prolong Live, for instance. There's also lipophilic cyanin dyes like Di C18 and Di-O that are membrane-based in terms of their binding and can be retained up to a day or two. Mm-hmm.
Okay, we have two more quick questions. Will Prolong glass mountant work with STED?
We haven't tested it in-house. We do have some collaborators who have tested some of our mountants for super resolution imaging, and it differs in terms of what's best for different methods, STED, SIM, PALM. The ideal mountant is different. I don't have in my memory here which ones were best. I remember Prolonged Gold at least was quite good for, I think it was SIM. But I'd have to double check that. Sorry, I don't know offhand if Prolonged Glass is good for that method.
That's okay. I'm going to do one more question before we move on, but there are several more questions in the chat if you want to take a look at them. Has Prolong Live been tested in bacterial cells?
No, as far as I know, we've only tested them on eukaryotic cell lines. My expectation is it would work pretty well for bacteria too, because it's basically sopping up the ROS in the media outside the cells. So whatever your cell type, it'd probably be good for those too, but we have not tested it.
Okay,
Thank
so
you very
there
much.
are more questions in the chat, so if you can look into that when we're finished, I'll go ahead and turn it over to Guillermo.
Oh, yeah. Thank you.
So Arvidas, are you introducing Kelly?
I think you need to be unmuted. we go.
Okay, so let me introduce Kelly Lonston. She is 18 years in the business of fluorophores and their applications. She is focused on emerging technology in the area of cellular fluorescent detection reagents, platforms, and analysis tools. She now runs her own consulting business called Luminous Bioanalytical Consulting, but previously worked at BioLegend and Molecular Probes in Vitrogen. Her training is in neuroscience, immunology, and the chemistry of the probe development. So I'm stopping my share, and so that Kelly can jump in.
Okay. I'm kind of new to my Mac, so hopefully you can make this work.
All right.
Is that good? All right. Okay, so thank you guys for having me. This is a very exciting topic right now. I would say that a lot of past applications that we all grew up with were often limited by the fluorophores available for the assay. And that is not the case anymore. We are absolutely in an explosion of reagent possibilities. You know, large Stokes shift dyes was an interesting topic because it really is the photophysical characteristic of a dye that allows us to do multicolor, period, whether it's in flow cytometry or microscopy. And so with a lot more of these tunable dyes, we're going to have more options on both sides to expand the palette of reagents for imaging and flow cytometry. So this is what I'm going to talk about since some of the physical characteristics have already been described. I figured I would just go through the gamut of all of the different kinds of fluors for fixed imaging and flow cytometry as well, versus what I think Luke will probably talk about, a little bit more live cell imaging and things like that. So 2 slides just on Stokes shift and FRET before we get into simple organic fluors like alexafluoroside dyes, DY dyes, etc. Protein-based fluors like PE, APC, PER-CP. Those are what we've always been using, right? Organic polymers, so things like the Brilliant dye family, the Super Brights from Thermo. And then organic polymers, DNA-based polymers, those are relatively new. Thermo just acquired the company Phytonics, so they're probably the most commercially viable for these. I hope you guys aren't distracted by my animals. There's going to be a lot of animals possibly in this talk, so whenever I start talking, they start getting active. So Sony also has DNA-based polymers, though, that I don't have a ton of data on, though. They've only released 1 fluor so far. And then QDOTs, which I'm sure a lot of you have already used in imaging, but you know, both Jason and I have supported QDOTs extensively over the last 20 years. And I always said exactly the same thing, which is that they were not a finished technology, that they were almost like absolutely a bandwagon that a lot of people jumped onto, but it was because there was such a pressing need to expand the palette of fluors. And so people would put up with a more cumbersome technology. And that's exploding, like with carbon dots, P dots, all sorts of dots of different matrices in order to control the size and the spectra of each of the dots. So I'll just touch on those at the very end of the talk. All right, so in terms of large Stokes, I kind of want to point out 2 different elements of it, not just the Stokes shift that we understand. So in this plot on the left-hand side, what we're looking at is an excitation at its peak at 645 nanometers and an emission around 699. That's what a typical Stokes shift that we would consider 100% efficiency on the excitation or the absorption side, which I always use those 2 interchangeably. And at probes, we were always chastised to understand the difference between absorption spectrum and excitation spectrum. Excitation spectrum being the photons, the molecules absorbing that will actually lead to the emission, which is the distinction. But then there's also the anti-Stokes factor. So you know, generally as biological imaging and biological applications are not the technology that is driving dots in general. It's all QLEDs, and they're always the electronics type applications, flexible tablets and things like that, that are driving the new fluorophores when it comes to light emitting diodes. So upconverting nanoparticles are one of those where basically you have to have a continuous amount of a lower wavelength energy before the particle can reach its proper excitation state. So it actually has the shift, it's a lower wavelength, it's a lower energy, higher wavelength excitation versus the lower wavelength, the higher energy emission. And that is the anti-Stokes shift. So I just wanted to bring that up because we are going to touch on upconverting nanoparticles. When you do tech services, as long as both Jason and I have in our lives, there are just 1,000,002 things that people do to their samples that can affect their spectra and the amplitude of the spectra, or the amplitude of the signal. Just 3 that came off the top of my head are pH, ion composition, and the proximity between the fluors. So, but it's always going to be these microenvironmental factors that immediately surround the fluorophore itself. For most applications, the fluorophore is surrounded by the antibody, so it's relatively protected from the rest of its microenvironment. But that's definitely not true, nor would you want it for a lot of for indicators. Any sort of chemical indicator that you're going to use for live cell imaging, it's not going to be protected like that by a bunch of protein, which is why we can benefit from sometimes its shift in spectra. Okay, so I'm sure we can all have 1,000,002 graphic captures from microscopy U because they have the best. So the 3, just to quick cover FRET, because that's really how we attain long Stokes shift. If we have organic fluors that have what we consider to be a wide Stokes shift or long Stokes shift, they're going to be extremely low energy because their quantum efficiency is going to be very poor. So they're not always very useful to us. The way that we have attained long Stokes shifts now is almost entirely by FRET, what we would consider long, which I'm going to kind of cover that too. So in this instance, you have to have one molecule that is able to donate an electron, and you have to have one molecule that's electron deficient. There can't be an exchange between them should there not be one that is capable of receiving the electron donorship. It's one of the reasons why I like this. So one thing that when people are looking, especially when I first started in tech services, a lot of people were trying to set up their own FRET assays without completely understanding that an antibody to an antibody is not necessarily FRET, because it depends on the localization of the 2 fluorophores and the distance between those 2 fluors. That's the FRET, not the biomolecule necessarily. Our tools are only reporting what we hope will be the distance at a certain angstrom between 2 things. And so that relationship between distance and fretting efficiency is something obviously that we can quantify. So the graphic I like so much on the bottom is maybe in a native conformation, the 2 fluorophores are too far away from each other to resonate energy effectively. But the minute that they maybe bind something or able to close in on itself, it brings the fluors within a certain distance of one another. So FRET is always the distance between 2 fluorophores, not the distance between 2 biomolecules. I bet you I called it fluorescence resonance energy transfer too. It's Forster. We all know it, and we all make that mistake anyway. So with simple organic dyes, the traditional Stokes shift that we're looking at is typically, it's small. Anything that we consider to be bright and useful in microscopy, we're lucky that these molecules shift, they're not just chromogenic. So we're looking at, around 25 nanometers worth of a shift in excitation to emission. If they do become longer than that, like the 3 that I have below, PAC blue, PAC yellow, and V500, they tend to be not very bright because of, when they jump to that excited state and they start to resonate that energy and they start vibrating, and they have different side chains on that molecule that can rotate or shift. All of that energy, that dissipation of heat and vibration is going to create that Stokes shift with that loss of energy. So it kind of inherently means it's never going to make the fluor brighter or more efficient by having a longer Stokes shift. That's why we have to rely on FRET to do it. So I talk about strengths and weaknesses all the time because it bothers me when anyone believes that something is perfect or that we're sold a perfect product. You know, everything is good for the application that it's good for. And we're lucky that it is. Pack Blue, Pack Yellow, and V500 got us 2 additional fluorophores in flow cytometry when we were just entering a phase of 8 to 10 colors. Everything was a step in front of the other to get us to where we are now at 26 to 40 colors for flow cytometry. So, you know, the strengths are that it allowed us to just start first making those steps, that progression towards multicolor. But the weaknesses are absolute dimness. All of these ones that I'm showing here are within a range of high autofluorescence, and they only have a practical utility for flow cytometry. So outside of simple organic dyes, we rely extremely heavily on phycobilly proteins in flow cytometry. If you're talking, so mostly we use RPE, some people have YPE. There's a few companies that are still trying to manipulate the sequence of these proteins in order to generate new repertoires of phycobilly protein related protein dyes. Same as has been done for GFP already. It's kind of the same idea. But you know, PE is a humongous molecule. It's 250 kilodaltons. It has, it's usually in a hexamer formation. So it usually has at least 4 leaflets attached, you know, that are combining to make a whole molecule. Embedded within the phycobilly, within the phycoerythrin, you're going to have, you know, around 10 phycobilly proteins, and those phycobilly proteins themselves are the phycobilins, sorry, the phycobilins themselves, they are the fluorescent molecule of that entire structure. You can't isolate them out and polymerize them. It's like phycobilly proteins were nature's first polymer in a lot of ways, because embedded within that protein are these single organic dyes. And that's what we're going after. That's what we're relying upon. So they have a lot of strengths, the largest of which is their Stokes shift. It's humongous. It also means they're relatively promiscuous in their excitation spectra though, which is not awesome for us in flow cytometry. What that does is it increases the spreading error, the distribution of our data when we plot them against each other. But the strengths are absolute brightness. I see you've got a cat there too, Guillermo. So the extinction coefficient's humongous. It makes it a great donor for a tandem. Because it's so large, when we do conjugate it to an antibody, it's in the hinge region of the antibody. It's the disulfide bond. And you can almost be guaranteed that the antibody is conjugated at a 1-to-1 ratio. So when you're trying to do anything quantitative, like not just single molecule detection in terms of single molecule brightness, but knowing for sure that you actually only have a single molecule attached to that protein or to that antibody, PE will get you that because it's so large that there's only room for one of it at the disulfide bond. So those I would consider to actually be strengths in a lot of ways. You know what you're getting. Weaknesses are stability, absolutely. It can't be made, you know, this is nature's polymer, not an organic polymer derived in an organic solvent. It's going to be extremely sensitive to organic solvents, things like ethanol and methanol, and to any sort of natural thing like heat that would degrade proteins generally. And then in terms of size, it can limit it can limit somewhat its permeability to small compartments or to the nucleus, but not typically in a way that bars it from that use in your assay. So in order to maximize that Stokes shift and take it a little bit farther, we'll make protein, you know, phycobilly proteins into protein tandems. PE-Sci5 is a really good example of that, although it's extremely photolabile. If you look at that single leaflet, those pink structures that they have, those are the embedded phycobilins, the chromophores or fluorophores themselves. That's what we're looking to get the Sci5 or the Alexa 647 or whatever the acceptor molecule of that tandem is. We're trying to nestle it in as close as possible to 2 to 3 of these fluorophores within that structure. So often what people will do is when they buy the phycoerythrin from, say, a vendor, they'll allow it to relax a little bit in a certain kind of salt solution. in order to try to allow penetration deeper into the body of the protein of the acceptor molecule in order to protect it from photobleaching and other physical factors, but also to try to increase the degree of labeling of that tandem. So that's actually a really important thing in flow cytometry, where we rely super heavily on these. A lot of people are like, oh, I get an entirely different product from different vendors. And you really do, because they buy all those materials and they make them in-house, and there are different, you know, methods of actually creating that tandem to begin with, let alone coupling it to the antibody. So the biggest strength of these molecules is that it made flow cytometry. The fact that we had these proteins at all and that we could make them into tandems, that's why multicolor flow cytometry became a thing. But their biggest weakness is going to be not only their physical stability, but the photostability of the acceptor. So fluorophores like Psi5 and Psi7, although they were also very potent acceptors of energy, they were also extremely photolabile. So now what you're going to see, since we're getting well beyond this point, is trying to create tandems generally that have a greater transitional energy efficiency, so that the energy that's being donated is almost as close to 100% as you can possibly get to being absorbed by those acceptors. That's number one, making sure there's very minimal spill back into the donor channel. And then second, increasing that total stability of the tandem, because the antibody itself already has its own limitations. We don't want the tandem to be so labile that, you know, it makes the assay less sensitive, basically. So they have exactly the same size issues as the normal phycobilproteins like APC and PerCP. It doesn't make it much bigger to have the acceptors on there, but it is still a very large molecule. I've always been surprised that the brilliant polymeric fluors didn't take off in microscopy more than they did. I think they're still wide open for them to really enable multicolor microscopy. This is actually my baby. This is what I did at BioLegend for 11 years. You'll see, basically, nature's polymer was a phycobilprotein. You have to have a ton of protein to encase these tiny little bilins, right? This is kind of throwing that on its head. Instead, you have an optical segment. You have a monomer that you can polymerize with spacers into a large, long polymer. But it's entirely organic chemistry. There's no lability like the protein, like PE and APC and PerCP. So you have a single optical segment that does not shift spectra when it's polymerized. There are enough spacers in between each monomer that they remain individually optically active. They don't homofret the way that you can see with a lot of organic dyes. So the length of the polymer is the thing that determines its brightness, but not its spectra. So the longer I make it, the brighter I make it. But then it also becomes extremely cumbersome because it is linear and it has a slight kink in its structure. So it becomes U-shaped because of how it's polymerized. So, and then you also, similar to PE, you have a base floor. So in this instance, it's going to be a shorter Stokes shift, one that maybe excites at 350 and emits at 380, or one that excites at 405 and emits at 420. You're going to have these base polymers upon which you build a family of tandems. This intellectual property is also extremely tightly held. So we're lucky to have the probes that we have right now until eventually some of that gets a little loosened and they can start to work on the chemistry again. So the ones below, BD owns the intellectual property on the brilliant dyes. Thermo has the super bright dyes, which I haven't tested nearly as much. So their strengths are an unlimited level of brightness theoretically, but in practice, that's something else entirely, because the molecule will get to be either too thick or too long to be useful for intracellular imaging or intracellular detection. But when I first started at Molecular Probes, there was only coumarin and Alexa 350 that was blue. It's why we used UXT and DAPI, because it was the only thing that you had. You couldn't phenotypically use these fluorophores for imaging. They just weren't bright enough. This allows us to have a brighter blue, basically. That's a humongous strength. I never thought that that would have been possible, and it isn't possible with basic organic chemistry. It's also very solvent stable because there's no protein in this polymer. It is not going to be as susceptible to things like denaturing solvents. But the weaknesses are a plethora because no technology period is ever perfect, and we have to accept its weaknesses with its strengths. It's definitely still a very large molecule. When it's conjugated to the antibody, you have a shorter stability time of the conjugated product. Again, I always say that anytime you're conjugating the hinge region, you're never going to make the antibody more stable. You're not going to increase the stability by going after the hinge. And that's kind of what you're seeing there. And also it has a propensity to non-specifically bind with itself. I call them uber brights. That's the technical term for that. But you start to see these extremely bright kind of single particle aggregates. And in flow cytometry, you start to see an inaccurate compensation, basically. And also they can't be lyophilized like other organic molecules can be. All right, so I would like to see more people imaging these dyes. The top graph there, or the top image there is actually from the original paper announcing the arrival of the brilliant violet dyes. It's really just comparing Dye-Lite 405 with BV421. It's kind of comparing apples to oranges if you really think of each antibody at its location and how many fluorophores are actually at that spot. It's just a massive amplification effect when you have a polymer like this. It would be like a difference between having 12 fluorophores reporting one antibody versus 4 fluorophores reporting one antibody. It's just an entirely different kind of ratio of signal. And when we think of photobleaching, it's a perceptual effect. If we already started out with a dim feature, photobleaching it, even if they were both at an identical rate, you would lose the signal faster visually with a dimmer fluor. So photobleaching is very much a perceptual issue until we reach the limit that we can no longer detect. Below it is a wide field imaging experiment where they just did it on blood, which is easy because when it comes to multicolor flow, we have all directly conjugated antibodies for immunology applications. That's not everybody else's reality. I would love to see some company come out with all these direct conjugates for neuroscience antibodies. That would be great. But so immunology, we already have, this repertoire of products that is not going to be at the disposal of most cell biologists. This one is a 6-color confocal imaging application with 4 lasers. This one, I think some of these are hilarious because they end up using like dyes that I would never, ever have recommended in a million years, like eFluor 710. I think it was PerCP, eFluor 710. Or they'll use PerCP or PE because of their big Stokes shifts. They'll basically just photobleach the first time that you lase them. They're going to be so sensitive to those high-powered lasers, you won't be able to store them and use them, to image them again later. If it was me, this is what I would like to see someone do. This includes a lot of dyes that are just recently being released, like the Kiravia Blue or the Brilliant Blue or the Star Bright dyes from Biorad. I have no commercial affiliation whatsoever anymore. I'm all game for everybody to make their impact in this space. But this is one that would make a very good combination if you were able to get everything directly conjugated. There are companies that do codecs. If everybody's familiar with codecs, one of the companies is Akoya. They use oligo tags as a hapten, same as biotin streptavidin. They use an oligo-oligo pairing between the fluorophore and the antibody so that you can use a non-species dependent secondary detection, because that's the limitation for microscopy. It's not the dyes anymore. It's the ability to take any antibody that you want, no matter what its quality is, and find a way to attach a hapten to it that's able to be selectively detected as a secondary detection, if not as a direct conjugate. So somebody needs to fix that problem. So this is an application that I did right away. So not new to microscopists, but new to flow cytometry, we have now entered the age of spectral detection in flow cytometry. And it's amazing. I never thought you could do it, just to be able to control the rate of the cell in order to collect enough photons to be able to unmix. Like, we know what it's like on a spectral confocal, you know, making sure that you have enough time that when you have two overlapping fluorophores, that you collect enough of each of their photons within that scanning time in order to accurately unmix them. It's one of the fundamentals of spectral confocal. And they now, this is the age of flow cytometry that we're at. So 26 isn't even, at the time that I did this a year ago, it was the most parameters that had ever been done. And now within like maybe 3 months ago, a 40 parameter paper was just published using all of the brilliant violet dyes, all the brilliant horizon dyes, and then a bunch of new dyes that use DNA as a backbone. But this was, the reason that we want to do this in flow cytometry is an application like what I did here. I want to be able to take any leukemic sample at all, AML, CLL, anything. And I want to be able to say, from the first time that I can see a progenitor, an immature stem cell-like progenitor, through all of its transitional states, in and out of different secondary lymphoid tissues, and down into my blood, I want to be able to know the state of affairs for that cell across the entire maturity spectrum. Post-COVID, it's going to be the age of vaccine memory. So this same sort of concept, but also for T cells, is going to be everything. We need to see all of them simultaneously. To only have snippets of them and then have to do a lot of parallel assays is not helpful, because you introduce a ton of error and just, yeah, you have too many variables when you start bringing it up into parallel panels. Okay, so there is also PDOTs. This is a recent release from Biorad, actually, although PDOTs themselves have been around for a very long time. It also is an optical unit. It's polymerized almost very similarly to how brilliant dyes and super brights are polymerized. An optical unit, some level of spacers, but how they fix some of the inherent problems of the technology is to lather it back on itself until it creates a dot. So it's still an organic polymer. It's just not a linear molecule. It becomes like an aggregation almost, but it's not an aggregation. Don't say I said that. It's the same idea. It becomes a dot. It becomes larger. It's about a 13 nanometer, like same as Q dots, between a 10 to 20 nanometer particle. So that can also be controlled though by how much of it you add. And again, the core doesn't determine the spectra the way that it does with Q dots. They're very bright. They've got a high extinction coefficient. They have all the same benefits as the organic polymers, but they don't non-specifically bind to themselves, which is a humongous thing for flow cytometry. They also can tend to have a higher efficiency of donating and accepting energy. So their fret efficiency is a lot higher, which is really important for us. We want to have very exclusively exciting dyes. But again, the weakness is going to be size. And right now it's just a limitation of testing. I would imagine they've got to be a little sticky. I don't know that for sure. I imagine they're going to have some penetration issues into thick tissue. Don't know it for sure. It's just all these question marks because there hasn't been enough testing in basic cellular applications yet. Okay, these are just some data that I got from Biorad, just showing brightness. One of the things that we need in flow cytometry is to replace the protein tandems because of their susceptibility to stability issues. So that is one place that this could actually be really helpful. Everybody wants to replace per CP-Sci 5.5, basically. Okay, these are the fun ones. So Jason's got to be happy that Phytonics just got acquired by Thermo. I know I would be excited. Such a cool technology. If anybody knows Eric Kuhl, this is the first time I heard of using DNA as a backbone. The concept is that normally we think of fluorophores as resonating energy on an XY plane, right? But that's not true, right? That's just because the fluorophore itself is planar. If you stack fluorophores like this, you can resonate energy through the column. So instead of having a polymer that becomes a long chain, you have its, a chain as in the planarity of the molecules is on edge with each other. Do you understand what I'm trying to say? Instead, it becomes a tube because the fluorophores are stacked on top of each other with the phosphate backbone, allowing that alignment of those fluors. So instead of a nucleoside, where the nucleotide has a phosphate on the nucleoside, take out that nucleoside and put in a fluorophore instead. And then you have space, 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 another fluorophore, space, 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 another fluorophore. So nature's second way of polymerizing dyes, one that we had to co-opt in order to create these dyes. When you look at Eric Kuhl's research, he's like trying to create the craziest spectra that you can imagine, where he'll have dye number 1, space, 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 dye number 2, space, 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 so that you have these emission spectra that are like fingerprints. They're crazy, but that's not helpful for us. We want exclusive excitation. Ooh, my watch wants to talk to me. You have exclusive excitation and you have defined emission. That's what we're looking for, is a no promiscuity. One of the ways that these molecules are also really interesting is that by knowing the sequence, you can create structures. It's called DNA origami. I can send you guys papers if it's legal, I can send you papers or give you references. How about that? But you can create all sorts of fun, anything you could make with carbon, you could theoretically create that structure with DNA. And that is what phytonics did. So instead of having these long strands, just singular tubes of DNA, they decided to use DNA origami to align the fragments of DNA that have embedded within them a fluorophore, so that they become aligned to create what they call a phyton, because that's what they're calling their particular shape. And then within that phyton, you can have donor and acceptor fluorophores. It becomes a highly tunable, highly selective, the ability for you to control the formation of this molecule is tight. And you can also make it as bright as possible because you know exactly where your fluors are going to be on the overall tertiary structure. You know where they're going to end up, so you can make them far enough away from each other that they don't quench. It's so tightly regulated. It's a beautiful thing. So like I was talking about before, what we're looking for is kind of a very focused, tight, an excitation that's only off of a single laser, and then an emission that has the tightest spread of the emission range possible. And that's actually what this looks like in flow cytometry on this cytometer, are these two graphs down here. It's looking just like in a spectral confocal, where maybe you have 10 nanometer increments across a certain spectral range. That's kind of how they see it too, just a little differently. So in a perfect world, you're only going to have emission into a limited number of channels versus it being wider. So they have gotten all the way up to 40 colors using the entire range of brilliant UV dyes, and they call them Nova Fluors. Sony also has a new technology that's very, very similar. It's not a phyton shape. It doesn't do DNA origami, but it does use that phosphate backbone to stack the dyes. I mean, it's just, these guys are just starting. Like for us to use these, they're gonna make microscopy explode, because you're gonna always have your acceptor and donor within a tight distance of one another that you'll always know exactly where they are. They have some interesting ways of displaying their data, because for us in flow cytometry, getting up to 40 parameters, that then becomes about data analysis. I don't want to do 2x2 parameter bivariates over and over and over and over. I may never find the thing I'm looking for if I do it that way. So we have to have just more intelligent algorithms to do data analysis. I certainly don't want to do what they did here with 40 parameters. That would take like a year. Some people may have already used quantum dots in imaging. I only have a few more slides, so I hope I'm not going too far over. Okay. Oh, Q dots. That was my entire last year at Invitrogen, was going to people's labs who were doing 18 color flow cytometry and conjugating their antibodies for them. I'm not sure if I'm ever going to have a love-love relationship with quantum dots, but They have an amazing extinction coefficient. They're just little semiconductor particles. They're going to keep absorbing energy, and they're just going to keep chugging along like a little motor, because that's really what they're good for, which means a high level of brightness, of course. And they have applications where they absolutely rock. If you're looking for infiltrating, okay, a great one is infiltrating T cells in a tumor. You may only have 4 of them in an entire image. You need something that's going to be bright enough on a single molecule level to see a rare number of things with sensitivity. That's a great application for QDOTs. Anything that's more punctate, where that amplification is exactly what you need, it's gonna kill for that. But their size is large. It's really best for cell culture, not so much for tissue. The organic solvents can hurt the organic layer that coats the dot itself, which will then hurt your antibody. So the image on the left-hand side here is looking at all the different sizes. When they say that QDOTs are 10 to 20 nanometers, that's the core and the shell. That's not the entire molecule. The entire thing with its organic layer and its biomolecules conjugated to that organic layer. Zinc sulfide is the shell of most of these QDOTs. There's no organic handles like a carboxylate group for them to be directly conjugated to that QDOT. You have to slather a layer of something organic around that in order to have amines and carboxylates available for biomolecule conjugation. So that's like such a big limitation. Any dots that come after it need to rethink how to get the antibody onto the dot. And it will really bring down the total size of whatever nanoparticle that they're using. So yeah, but like I remember one of the first applications where, well, you could have a 20-year-old microscope sitting in the corner of some lab, and as long as you can change out the filters to see these emissions, all you need is that single wavelength of excitation, and you could have theoretically 6 wavelengths of emission if somehow the dots were compatible with your biology. That wasn't really always the case though, but they have a lot of strengths. I would love to see new generations of QDOTs come out, which is kind of what the next slide is, and the last slide that I have for you guys. Actually, I think I have one more, is using other types of fluorophores like lanthanides or upconverting nanoparticles. I think there's more of a future in C-dots or carbon dots, carbon nanoparticles. You can dope those lattice structures with simple organic fluorophores similar to how they're polymerizing the DNA and the other organic polymers. You're going to be able to control and tune a carbon dot tighter without influencing the size or the shape of the lattice structure. And then from those, you're going to be able to make tandems. And you're going to miraculously go back to being able to get these long Stokes shift dyes that we all need to do the interesting work. So this was just out of one paper where they were looking at some single molecule imaging of the different kinds of molecules. Here they call them C-dots, and Q-dots are at the top there. And I would agree, like the potential for these molecules to become brighter and to become more tunable, to be under our control, is one of their biggest promises. So this is just giving some credit where credit's due, because, we may work, well, I don't work for a commercial vendor anymore, but, I have my entire life. There's so much research that goes into probe development that is constant. I highly recommend reading ACS journals, because that's really, where the fundamentals are developed. And then eventually they need to get to somebody that's going to be able to commercialize them and take them to a biological setting. So I think it's worth giving a shout out to all the manufacturers, and my contact info is there. I do consulting now on all sorts of bioassays. So feel free to come to my website or to contact me directly.
Right, Kelly, thank you so very much. We are at 2:07, and people may have other things to do that they have scheduled. So I think we are going to punt your Q&A to Tuesday. Is that okay with you?
Yeah, it's fine.
Thursday.
Okay. So because I know that I apologize for running late. I realize that some of you have other things to do. attend. We'll be reconvening on Thursday at 1 p.m. Eastern, Thursday the 25th, and hopefully we'll have the PDFs posted ahead of time for you. Thank you very much.
Thank you, everybody.
Thank you.
Hi, Aaron. Can you stay on for a quick second? It's Ken.
Yeah, sure.
Sorry about the polls. Did I miss your signal? Did you want them to run automatically?
You did not miss my signal, but I missed your confirmation that the polls
Oh.
are available.
I'm sorry.
So
I'm
this
sorry.
is why. So I'm wondering if we could run them on Thursday at the end.
Whatever you think.
Yeah,
It's all set, ready to go.
that's very good.
Because there is, for me and for many people, first try. So like it takes time to figure out how it
Right.
is organized.
Okay.
Cool. Can we save the chat so we can keep the questions and all that for Tuesday? Revisit this thing.
Mm-hmm.
Okay.
Yeah, it'll download with the recording. It'll download, and I can send that on.
Okay, that's good.
One more question, guys. Was my pointer visible during my presentation?
Don't think so.